Swayam Prabha. Digital India. Educated India.
In previous lectures, we have discussed about the basic principle of electrophoresis and various techniques concerning the electrophoresis of proteins and nucleic acids. Today, in this lecture, we are going to discuss about two more techniques. One is immunoelectrophoresis and another is the capillary electrophoresis. So, we will discuss immunoelectrophoresis first. Now, immunoelectrophoresis is a technique for studying antibody and antigen, particularly their interactions, where the specificity of immunoprecipitin reactions are combined with separation of molecules by electrophoresis. So, this technique allows you to see the interaction or to identify the antigen if the antibodies are available. Now, before we go into the technique of immunoelectrophoresis, certain basic things needs to be understood. Like we were talking about immunoprecipitin reactions. Now, what are these precipitin reactions? So, let us understand this. Now, precipitin reactions, supposing if increasing amount of an antigen, say human serum albumin or some other protein is mixed with a suitable fixed amount of antibody solution, say rabbit anti-human serum albumin or for that matter against any particular antigen, what will happen initially the amount of antibody antigen precipitate increases as you increase the antigen concentration. Now, as you are adding, what is happening is one thing has to be understood is many times there are two sides on the same antigen where the antibody can bind. It means you have more than one antigenic determinant. So, for example, there are a lot of repetitions or certain proteins, membrane proteins or receptors which are present. There are specific repeating features which are present, which can act as antigenic determinants. So, when antigen interacts with the antibody, antibody can bind to two antigen molecules. And what will happen is if there is certain concentrations of antibody antigen at particularly at equivalence, then they form a large lattice sort of structure which leads to visible precipitation. It means that the structure becomes so large that you can see it as a precipitate. I will show you that, and that precipitate is called precipitin. So, in precipitin reaction at certain point when you add antigen to a suitable amount of antibody or a fixed amount of antibody, at a certain point when concentration of antigen and antibody reaches equivalence or equal, then there is a lot of interactions and a large lattice structure forms which leads to the visible precipitate. All right. So, now when you are adding, I will show you this in a little while. So, as you add the antigen and as I said a lot of precipitate is formed and visible precipitate which could be seen is formed, but what happens is as you keep on adding the antigen, a sharp plateau indicating that all of the antibody had been precipitated is not obtained here, and rather precipitate apparently dissolves at higher concentration of antigen, and this is due to the solubility of antigen antibody complex containing a single antigen molecule. Even if the antibody molecules is bound to every antigenic determinants, the resulting precipitin curve will contain. So, what is happening here is that at excess antigen, the complex which is visible precipitate will dissolve. Now, so if you see the dynamics of this or precipitin curve as we can call it, it contains 3 zones actually. One is zone of antibody axis, where the addition of antigen leads to a substantial increase in the amount of precipitate. So, as you start adding the antigen from a small quantity, then there will be antibody axis. Second part of the curve is zone of equivalence, where when the maximum antigen antibody precipitate is formed, and third part of the curve is the antigen axis, where when you add more antigen, that is after equivalence, the precipitate dissolves actually. All right. So, let me show you this whole thing on the screen. What you have is, if I show you the precipitin curve will look something like this. You have antigen on this axis and you have antigen antibody, Ag for antigen, Ab for antibody precipitate, or you can say precipitate which is kind of seen as you add the antigen. So, what you see is there is a curve kind of looks like some sort of this kind of pattern, where you have certain regions actually, and these regions, if we call them, this one, that is the first one, is the zone of antibody axis. This is antibody axis. This is zone of equivalence. And here, when you have added sufficient quantity of antigen, then it is zone of antigen axis. So, what does this means actually? This means, like, if you see in first set you will have lots of antibodies which are around, and there will be certain interactions, but and as you are increasing this there will be antibody axis, but as you increase the antigen there will be some precipitate will be seen, and on increasing concentration of antigen, visible precipitate will be more. Now, in zone of equivalence, large lattice structures are formed. Like I said, if you have a lot of these equivalent antigen molecules, it's like you will have a lot of these kinds of structures which kind of are in a long range, and they will be formed in a very long range, and they will be like visible precipitate which could be centrifuged and measured also could be obtained. In zone of antigen axis, you will have a lot of antigens as compared to number of antibody molecules, and there could be like lot of antigen which is because you have a fixed amount of antibody. So, this is a curve, or we call it precipitin curve. Now, how do you see this curve as you go along? There are a lot of different methods to see this curve, and there is one process through which you can see the antigen antibody interaction is immunodiffusion. So, there could be different forms of immunodiffusion, which could be single immunodiffusion and it could be double immunodiffusion. Single immunodiffusion means that only antigen is moving and antibody is immobilized, and it could be in one dimension or it could be in two dimensions. In double immunodiffusion, both antigen and antibody moves towards each other and interacts to form the precipitin lines, and that could also be in one dimension or in two dimensions. So, let us see that the phenomenon of immunodiffusion. Let me show you on your screens actually. All right. So, the immunodiffusion, one is, like I was saying, it could be simple immunodiffusion or single immunodiffusion, where only one one of the reactant, that is antigen, moves towards antibody. It could be, like I said, it could be in one dimension or it could be in two dimensions. So, say in one dimension, if we are saying, if this is particular, now if this is your agar containing, this is your agar containing agar having antibody, and this is antigen solution kept in here. Now, so antigen solution is kept on top, and this is agar which contains antibody, a particular antibody for that antigen at time t0. Now, these things takes immunodiffusion takes a lot of time, like days together, and so we will not be talking about that, but what we are talking about is simply the phenomenon. So, what happens is, what will be the second step in here? All right. So, it is the same thing. Now, this antigen solution and this is agar containing antibody. Now, what will happen is, slowly the antigen will start moving into the agar which contains antibody. Now, the precipitin line or the curve which I have shown you, as the antigen enters, there will be antibody access. So, what will happen, as antigen interacts with the antibody, there will be a very minor precipitate is formed, and there will be very thin precipitin line could be seen. Now, as antigen enters, more antigen enters into the gel or agar, then slowly the antigen concentration increases, and then from antibody axis you will find antigen antibody equivalence at certain point. So, what will happen, first you will see a precipitin line as a visible, like, because it will be very thin in the beginning. So, you will see a precipitin line, and this precipitin line is seen as antigen concentration increases. Now, what happens is, the precipitin line moves in this direction actually. The precipitin lines moves down here, because as the antigen comes in, antigen axis occurs, and at certain point the precipitin or antigen antibody complex dissolves. So, antigen but antigen concentration ahead is little less. So, what happens is, essentially the precipitin lines starts from here, but it keeps on moving downwards. So, what you will get is, finally at certain period of time you will see that the anti-precipitin line has moved quite far depending on the antigen concentration, because after some time antigen will be done, and so precipitin line will move to a certain extent. So, you can see this at say time 1 at 3 different times, say, and this is in days actually. You will see that precipitin line is formed, and then it moves down, and at certain point it stabilizes, and that depends on the concentration of antigen. So, that is how a precipitin lines can be formed and can be seen in single dimension. Now, this thing, simple immunodiffusion, can be in 2 dimension also. Now, how does it happens in 2 dimensions? Now, what is done is, in 2 dimensions, this is done on a very simple microscopic slide actually. Now, here what you do is, you punch different you can say holes or slots for loading the antigen solution. So, these are the wells which are being cut, these are wells actually, and these wells have been cut in here for putting in the antigen solution. Now, here different concentration of say 50, 100, 150, 200, increasing concentration of antigen is loaded in here, just to see the effect here. So, what we will see is that there will be radial diffusion in all directions in all the wells once you have loaded the sample. So, it will but it is in simple diffusion, since only antigen is moving, and this agar this antibody is contained in agar actually. So, antibody with agar is put in here. Now, when the movement occurs, then what will happen? Precipitin lines will be formed in here. Now, as the concentration increases, so is the size of the or the diameter of the precipitin lines will be. So, what you have seen at 50 micrograms, say for example, just for example, there is a precipitin lines which is not farther from the center here, and as the concentration increases, the precipitin line also moves. So, this clearly shows that more is the antigen concentration, farther the precipitin line will be formed. As we have seen in here also, simple simple immunodiffusion also in one dimension. So, what you can do is in here, you can always make a standard curve here, and this standard curve can be used for knowing the concentration of unknown sample. So, this is simple immunodiffusion, and you can get precipitin lines which can be visualized. All right. So, this was simple or single immunodiffusion. Now, same thing can happen with double immunodiffusion. Let us see how double immunodiffusion will occur. In double immunodiffusion, what you are going to see is that rather than simple this antigen moving, both things will move. So, what you have, if it is a single dimension, in single dimension what they will have is there will be antigen solution, and then there will be agar which does not contain anything, and there will be agar containing antibody. So, antibody in agar. So, here what will happen? Here this is not immobilized antibody, with the antibody in here is not immobilized. So, what will happen? Antibody will move towards this side, and antigen will move in towards the antibody, and antibody will move towards the antigen. So, what is going to happen now? That as it moves, as both things move towards each other, you will get, and as we have seen, you will get lot of precipitin lines depending on. So, these are what you call precipitin lines. Now, you will see one precipitin line if there is only single antigen and antisera for that. You will see multiple precipitin lines if it is a complex mixture of antigen and corresponding antibodies are available in that antiserum. So, this is double immunodiffusion, that is in single dimension. Now, this could also be in, like I said, two dimensions. In two dimensions what is done? There is a very simple way, like there will be agar on this microscopic slide. You can punch a central hole where you can put antiserum, and you can have slots for placing antigen, and these antigens could be in different concentration. Now, depending on the concentration, like I said, more is the concentration of antigen, farther will be the precipitin lines. Accordingly, you will get a pattern which will be seen in here, that is precipitin lines will be seen as per the concentration, and this is double immunodiffusion. Here antigen, like these are moving in all directions, likewise antibody is also moving in all directions, and at certain point they will be in equivalence and where they will form a visible precipitin lines. So, that is the double immunodiffusion. Now, there is another important part of this double immunodiffusion is that how does they look like there are like reaction of identity or partial identity or non-identity are very important. For example, if you have same antigenic determinants, what happens if there are partial resemblance in antigenic determinant or there are no resemblance in antigenic determinant. So, let us see what is that. Now, everything is same, like you have a plate, microscopic plate, you have a agar containing things. So, what is done is to see this particular phenomena, that is reaction of identity. One is that you have, like I said, like here it is antigen and there it is antibody, that is anti AB. Now, see here both have same antigenic determinant AB AB. So, what will happen? This is reaction of identity, and how does this particular precipitin line will look like, that what we are interested in. So, precipitin lines in this case will kind of merge actually, that is the precipitin line of interaction between these two and precipitin line of the interaction between the both wells, they will merge with each other, and so this is called reaction of identity, where you have identical antigenic determinants. Second case could be where you have non-identity, that is both antigenic determinants are. So, second one could be reaction of non-identity. Now, in this case, like I said, they could be different antigenic determinants, like say I say these are two antigenic determinants, you have only anti AB. So, what you have is you do not have common antigenic determinant. So, what will be the resultant? You will find a kind of a pattern which crosses each other, they are not identical. And third case could be partial identity, the reaction of partial identity. In partial identity, there is at least one common antigenic determinant, like say if I say this is AC and AB and you have anti AB same antibody or antisera, then what will happen is, since there is only partial identity, it will look something, this sort of pattern will be seen, that is in case of that at least one antigenic determinant is common. So, this is how this will look like. There is another important or interesting aspect of immunodiffusion in here, and that is you can determine or you can have an idea of the molecular weight of a particular antigen, that is effect of molecular weight of an antigen on the precipitin lines actually. So, that is also very interesting to see. All right. So, what we can, let me show you this here. All right. So, again this is a microscopic slide where agar is there and you have punched two holes. So, let us see first in the case of this is antigen, say this is antigen A and this is anti A, that is antibody A or anti serum you can say against antigen A. Now, to just like it will not give you say what is the molecular weight, but it will give you an idea about the molecular weight in terms of whether it is bigger than antibody or it is smaller than antibody. Now, in case of where antigen has a smaller molecular weight as compared to antibody, then what you get? You get a pattern something like this, that is you have like a pattern where precipitin line will be towards like it will curve towards the antibody. There could be another one, that is say antigen B and there is anti B. These are the slots we have in agar plate, this is agar plate and it facilitates the diffusion because it is a large pore size is there, it is not a separation as such. Now, say antigen equals antibody, molecular weight equals, then you will see a straight line here rather than a curved line. And if you have, I think you must have guessed the third scenario, say there is antigen C and there is anti C. The third scenario would be that if say antigen is bigger than or antigen molecular weight is more than the antibody, then there should be curve which is different or opposite to the curve which where antigen molecular weight was lower. So, these kinds of patterns will be seen and could be give an idea on in this particular experiment of immunodiffusion. All right. So, let us go back to our discussion. So, we were discussing about immunodiffusion and precipitin lines. Now, let us get back to the immunoelectrophoresis. So, immunoelectrophoresis is extension of what we are discussing, and it could be two kinds, qualitative and quantitative immunoelectrophoresis. Now, first step in immunoelectrophoresis is to separate antigen sample into their component part by electrophoresis. So, that is why it is a immunoelectrophoresis, because you are performing electrophoresis to separate the mixture of antigens on agar gel. In second step, it will be similar to immunodiffusion. What is done is these separated components are probed with antibodies in the gels, and the specificity of antibody facilitates the identification of antigen in given sample. So, what is done is a very thin, say 1 to 2 millimeter agarose gel, it could be say around 1%, gel is cast on a glass plate or microscopic slide, and antigen samples are placed in small circular wells, as I have shown you, which could be 2 to 4 millimeter in diameter. It is punched in the gel. Now, gel is placed between the electrodes chambers in horizontal electrophoresis setup. Now, mostly it is the anodic migration, that is from negative to positive electrode. Now, gel is in contact with the electrophoresis buffer through buffer saturated filter paper wicks. A suitable immunoelectrophoresis buffer, which can be like 0.08 molar tris, 0.02 molar tristin, 0.3 millimolar calcium lactate pH 8.6, or there could be other buffers will be utilized. And the electric field is applied in order of say 5 volt per centimeter. Now, what is done? Like it is a simple electrophoresis. After electrophoresis is complete, then antigen mixtures will be separated as per their charge. Then antisera will be placed in longitudinal troughs, which are 1 to 2 millimeter wide. I will show you this, which are again cut into the same gel and parallel to the migration path, and one trough is cut on each side of the sample lane. Like if you have 2 sample lanes, then you can cut 2, if it is a 1, you can cut 1 trough, and different antisera will be placed in there. Like if there are 2 lanes or 3 lanes, then you have to have antisera for each. Now, gels are then incubated overnight at room temperature in humid chamber. Remember, all these experiments of immunodiffusion are done in humid chamber. The antigens diffuse readily and antibodies will diffuse laterally, because they are in longitudinal troughs towards each other, and resulting into antigen antibody interaction and precipitin arcs, where antigen and antibody have appropriate concentrations, that is equivalence actually. Now, the precipitin arcs can be visualized directly, or they could be seen by staining, like say presence of precipitin arc is evidence for both antigen in the sample and the antibody in the antiserum. So, this is how simple qualitative immunoelectrophoresis will be performed. Alright, so let me give you an idea about how the whole procedure is being done, but as we have already done this in earlier, just to give you an idea. So, what you have is a simple microscopic or glass slide, and this glass slide contains, if I say particularly, what you have is, and you have say particular antigen sample, particular antigen mixture. Now, there will be a longitudinal trough in here, which is cut in the gel only. So, what will be the next step? Next step would be the electrophoresis. In electrophoresis, what will happen? You have so your sample will run. Now, remember, since it is from cathode to anode, those samples which carry a negative charge will run towards this side. So, that has to be experiments have to be planned accordingly. So, when it runs, what will happen? There will be a migration, and you will find as it runs here, you will find different bands in here, that is separated antigen from components from the mixture. Now, what So, once you have run the electrophoresis as we have discussed, the next step would be to fill this. So, next step would be, so what you have is you have these components from antigen mixture and you have what you will fill in anti-sera for that particular or antibody solution for that particular antigen, and this contains antibodies for each of these samples, or for one sample, like say you are looking for to identify certain sample, then it might contain antibody for that. So, what will happen now? There will be lateral diffusion like I said from this well, and there will be radial diffusion from these in all directions. So, what resultant would be? You will find after some time, like say incubating overnight, you will find them. So, what you have is you have these kinds of patterns, or and you have anti-sera here. What you will find is you will find the precipitin lines for these samples actually after incubating overnight. So, you will be able to if it is to identification, then you will be able to identify, because you will find only precipitin line for that particular antigen sample you are looking for. So, this is a very simple qualitative immunoelectrophoresis. All right. So, that is how you can perform immunoelectrophoresis and could identify or you could analyze the particular antigen. There could be quantitative immunoelectrophoresis also, 1 and 2 dimensional rocket immunoelectrophoresis. Now, the two major variations of this is 1 and 2 dimensional rocket electrophoresis or immunoelectrophoresis, and the name rocket is due to the rocket shaped precipitin patterns formed following antigen antibody reaction. Now, height of the precipitin peaks are roughly proportional to the concentration of antigen in the sample, and therefore, these methods can be considered as quantitative immunoelectrophoresis. In rocket immunoelectrophoresis, antigens are subjected to electrophoresis as we have shown you in agarose gels containing suitable antibodies, and the pH of the electrophoresis buffer is chosen so that it is near to the pi of the antibody molecule. So, that antibody remains immobilized in the agar or during the electrophoresis. Now, for one dimension rocket immunoelectrophoresis, antigen samples are loaded in the well punched in antibody containing gels before starting the electrophoresis, and what you will get is you will get rocket shaped precipitin peaks where antigen and antibody meet at equivalence. Now, the concentration of specific antigen can be determined by comparing rocket height of test samples with those formed by known standards. Now, in 2 dimensional rocket immunoelectrophoresis, what is done is first step is to perform regular agarose gel electrophoresis to separate the antigen in the sample, and in the next step the lane containing the separated antigens will be cut like we have discussed in 2 dimensional gel electrophoresis, and will be fused to an agarose gel containing embedded antibodies. Second electrophoresis will be run at 90 degrees to the first one, and then again you will get the same patterns as the antigen migrates, that is rocket shaped pattern. So, the precipitin arcs are formed along the equivalence regions of antibody and antigen, and number or multiplicity of the precipitin arcs formed will depend on the complexity of the antigen sample and the anti-serum in the second gel. Let me show you this on your screen. So, two ways this could be done is, one is you have agar containing gel, antibody containing agar or antibody in agar, and this is antibodies are all over they are when you are casting the gel you put the antibody. Now, what you do is you punch holes in here or slots for loading your sample and unknown, and as you run the electrophoresis in one dimension, then you will what you will get is, say we have loaded the unknown in a particular concentration, say 5, 10, 15, 20 or so. So, what you will get accordingly the antigen concentrations will get rocket shaped pattern, and like in chromatography this particular heights of the peak will give you the concentration idea. And so, unknown could be found out. If unknown is this height, then you can through calibration curve you can know the unknown. So, this is called rocket electrophoresis because of this pattern. Now, in two dimensional rocket electrophoresis, what is done is that first it has been done in this direction electrophoresis, that is you have separated these components here rather than punching hole, and this whole thing has been fused into this next gel and which contains the antibody. Now, as they run in this direction, now direction of run will be this one, that is at 90 degrees, and you will get the patterns which will be, like I said, it could be different patterns because of there will be a lot of mixture. As complex is the mixture, the pattern will be that complex. So, two dimensional rocket immunoelectrophoresis could be performed, or it could be one dimensional immunoelectrophoresis. Both ways it could be performed. Alright, so once precipitin lines are formed in immunoelectrophoresis, they could be visualized. They should be like I said, this should be precipitin lines should be developed in humid chamber, and they could be directly seen by naked eyes, and if not, you can illuminate it from the side, and on a dark background they could be seen clearly. Many times staining might be required. They could be stained with regular protein stains, and then they could be like excess thing could be washed off and they could be seen. They could also be permanent records can be made by photography and other means. So, this is all about immunoelectrophoresis. Let us move on to the next technique, that is capillary electrophoresis. If you could recall, we have talked about a phenomenon called electroendosmosis, or this particular in isoelectric flow actually. In electroendosmosis, there is a net cathodic migration. So, capillary electrophoresis works on or takes advantage of that particular phenomenon. So, capillary electrophoresis could also be like, it is also known as capillary zone electrophoresis, and there are other names. It separates the ionic species by their charge, and then other factors could be frictional force and hydrodynamic radius, but mostly on charge. Capillary electrophoresis involves electrophoresis of samples in a very narrow bore tubes, fused silica or other, and these are like say internal diameter could be 50 micrometer and external diameter could be 300 micrometer. So, these are very narrow bore fused silica tubes. Now, one advantage of using this capillaries is that they reduce the problems resulting from heating effects. So, because this particular surface to volume ratio is there is large surface to volume ratio and heat could be dissipated easily. Now, but it does not mean you can keep any length to do that. There is certain balance between the length and the voltage, and these are run at high voltage. So, high voltage will generally cause lot of heating effect, but since they are long and thin bore or narrow bore capillaries, therefore, the heat dissipation can take place. But I said like increasing the capillary length will not improve as such the separation here. Now, types of molecules that can be separated, there can be different kinds of molecules can be separated by capillary electrophoresis. They can range from say protein, peptides, amino acid, nucleic acid, inorganic or organic molecules, organic acids, and lot of other different kinds of molecules could be separated on capillary electrophoresis. As we have talked about this particular phenomenon of electroendosmosis, the inside wall of the capillary is covered by, if you could remember, we have discussed about this, that is silanol groups which are charged above pH 3 or starts charging after pH 2, and this negatively charged groups attracts cations to the inside wall of the capillary. Now, what will happen? The distribution of charge at the surface is like there is a stern double layer which is like kind of fixed, and there is a diffused layer, and this results in zeta potential. Now, zeta potential is the potential at any given point in double layer and decreases with increasing distance from the capillary wall. So, it will be more at closer to the wall, and the Stern model is also referred to as electric double layer model. So, what is happening here is, you can see here there is a fused silica capillary tube, it carries negative charge, and negative charge will increase as the pH increases. Now, what you have seen here, there is a fixed, or you can say in this figure, rigid layer, and there is a diffused layer, Stern layer actually. So, rigid layer is very close to this, and diffused layer has some leeway to move around. So, the electroosmosis or electroendoosmosis flow is utilized in capillary electrophoresis, and the separations occur due to this phenomena. So, what happens is that in electroendosmosis, the charges or the positive charges which is in diffuse layer moves towards the cathode and carries the whole solution with it. So, there is a net flow towards cathode. So, net flow occurs as solvated cations drag along the solutions. Now, in this figure, as the pH increases, the mobility also increases here, and that is because the charges increases in here. So, net flow is towards the cathode, and it carries everything towards cathode, that is even negative charges entities towards cathode. Now, if you consider like say there is a 50 millimolar pH 8 buffer that will flow through a 50 centimeter capillary at say 5 centimeter per minute speed with 25 kilovolt applied potential. So, that is how it will flow, and key factors that affect electroosmotic mobility will be dielectric constant and viscosity of the buffer. Now, this is like, so if you see here, since they are all moving towards one side, many times it is hard to really separate neutral or negatively charged species as you want to. Many times electroosmotic flow could be quenched by say protection of silanols or low pH. You can coat the inner wall of the fused silica, and so to quench or to suppress the negative charge, but then problem will be that only one kind of ions will move towards the cathode, because net flow or your detector is towards the cathode. These equations here give you an idea about the electroosmotic mobility, and which are dependent on dielectric constant of the buffer, zeta potential, viscosity, electric field, all these factors affect the mobility. Now, a typical capillary electrophoresis, a very simple schematic is shown in here in this figure. If you see here, there are two reservoirs, and this is anode, this is cathode, this is anodic reservoir, this is cathodic reservoir here. The capillary tube is dipped in here, anodic and cathodic, and there is it could be 10 to 100 micrometer inner diameter plus 20 to 100 centimeter length could be taken. There is a high voltage supply which could go up to say 50 kilovolt, but around 25 kilovolt is utilized. There is a detector at cathode side which can detect or which will be monitoring the whole run, and in a recorder, data recorder, these things could be recorded and analyzed. So, this is a typical capillary electrophoresis setup, and the sample is loaded towards the anodic side. We will see how. So, what has happened is systems like as we have seen, main components are sample vial, source and destination vials, capillary, electrodes, a high voltage power supply, a detector, and data output and handling devices. The source vial and destination vial which I have shown you, these are the source vial, anodic one, and the destination vial. They will like here these are filled, and capillary also is filled with an electrolyte which is aqueous buffer solution. Now, to introduce the sample here, the capillary inlet. So, what will be done is that anodic buffer chamber could be removed and the sample chamber or sample containing device, the say it could be a simple syringe type or a beaker could be put in here, and sample could be loaded or introduced into the capillary by different ways like capillary action. It could be a pressure injection where you can through a syringe, it could be by pressure you can put in sample, a small amount of sample, or it could be high voltage injection where you replace the anodic chamber by sample and very instantly for a very small time high voltage is applied, which leads to the introduction of sample into the anodic side of the capillary, and then again the capillary could be returned to the anodic chamber. Then the migration of the analytes is then initiated by an electric field that is applied between the source and the destination vial, that is anodic and cathodic chambers, and is this is like supplied to the electrodes by a very high voltage power supply, as I have shown you in this case. Alright. So, it is important to note that all ions, that is positive as we have discussed earlier, negative. So, positive, negative or neutral ions will be pulled through the capillary in the same direction by electroosmotic flow. Now, the analytes separate as they migrate due to their electrophoretic mobility and are detected near the output end of the capillary, that is cathodic side. So, output of the detector is sent to a data output and handling device such as an integrator or a computer where it will be analyzed. Data is displayed as an electrophoretogram which reports detector response as a function of time. Now, separated chemical compounds appear as peaks with different migration times in an electrophoretogram. So, in electrophoretograms, if you see, like we said, detectors are placed at the cathode side under common conditions, and these detectors should have high sensitivity because the speed at which these are gone is very fast. Now, if you see the electrophoretograms, what you will see is the cations will come first because they are obviously moving towards cathode and they will move faster, and depending on how much charge is present, like say if some analyte contains more positive charge, it will move faster, then neutral will go will be pushed in, and then anionic, because anion will also have electrophoretic mobility that will push them or move them towards the anode, but since net flow is towards cathode, they will be the last to appear there. So, that is how the different analytes will be separated. There are a lot of advantages of capillary electrophoresis. In advantages, it offers new selectivity and an alternative to HPLC, though HPLC is more widely used. It is a very easy and predictable kind of selectivity it has. High separation efficiency, that is you have high theoretical plate number. There is a very small sample size is put in. Separations are very fast, like say 30 to 40 minutes or maybe less. It is automated, it could be quantitative also. It could be easily coupled to say mass spectrometry or others, and lot of different kinds of modes are available, like as we will see. There are a lot of disadvantages also. It cannot be preparative, like you cannot do very large scale preparations or separations. Low concentration and large volumes are very difficult to do. Then there are problems of sticky compounds, like for example, certain proteins might stick to the negative charges, positively charged, and there could be streaking of the protein and you will not get a sharp peak. There might be certain species which are hard to dissolve. Reproducibility is another problem. It is very hard to get many times reproducible results. So, these are certain disadvantages, but there are many advantages to use capillary electrophoresis. There could be many modes of capillary electrophoresis, like say there could be capillary zone electrophoresis, where in free solutions you are doing or free buffer you are the experiment is being done. There could be capillary gel electrophoresis, where a particular matrix material is loaded or matrix material is packed in these columns. Again, it is very hard to do it by oneself. It is like commercially available columns are available. These are commercially available columns, and they can it is like by different strategy the gel is put in there. Isoelectric focusing mode could be available, and for neutral molecules there is micellar electrokinetic capillary electrophoresis or chromatography we call it, where a detergent could be used and a neutral molecule could partition when these micelle formation of detergent at certain concentration takes place. So, these are different modes where could be utilized in capillary electrophoresis for different applications. There could be many applications of capillary electrophoresis. It is a very versatile technique for a lot of different kinds of compounds to be separated and analyzed, though certainly instrumentation and it is an expensive instrument, but it is very handy. Lot of different kinds of like pharmaceuticals, in pharmaceutical industry it is utilized for say reaction intermediates, purity validation, stability, final product testing, ion analysis, and lot of other different applications. In biosciences, peptides, proteins, DNA, carbohydrates can be analyzed. In food industry, inorganic cations, anions or organic acids, amino acids etc., could be analyzed. In environmental science, lot of pollution studies or pesticides or lot of other different kinds of metals, surfactants, dyes, environmental pollutants could be analyzed on capillary electrophoresis. Then forensic science, like say drugs of abuse, explosives, gunpowders and other things could be utilized in could be analyzed in capillary electrophoresis. So, lot of different areas utilize capillary electrophoresis for different applications. So, this was about capillary electrophoresis. Another very important technique which could be utilized for a particular applications by various kinds of in various kinds of fields, actually various fields of biotechnology. So, this completes our section on electrophoresis. In this lecture, we have discussed about immunoelectrophoresis and the capillary electrophoresis. Both are very important techniques. Immunoelectrophoresis has been nowadays replaced by immunoblotting techniques, where like in Western blotting or others, the particular molecule, say protein molecule, can be transferred on nitrocellulose membranes and can then be probed by antibody linked to an enzyme, and then it could be the bends could be or the patterns could be seen with enzyme reactions. So, this has but still immunoelectrophoresis is utilized for various applications. Likewise, capillary electrophoresis is very good, very simple to use automated technique for various applications. In the next lecture, we will start a new topic which is also very important, that is centrifugation techniques. Thank you.